Hello and welcome to our lecture today, talking about the differences between G protein linked receptors and the intracellular and the first messenger system. So we're going to be comparing first messenger system to G coupled protein systems, which are a division of the second messenger systems. So in order for the message to be relayed and to get a target response, we need a messenger. In the case of steroid hormones, the messenger will do the job. It will leave this messenger, this steroid hormone will be the first messenger. It will leave the blood circulation and then exit the blood through these interendothelial gaps and then swim in the interstitial fluid, then cross the bilayer leaflet of the cell membrane, then swim in the cytoplasm, and either in the cytoplasm bind a receptor, then mobilize to the inside of the nucleus, or cross the nuclear envelope and then bind the receptor that is in the nucleoplasm, and then mobilize to the DNA and then transcribe for us a target mRNA, and then this target mRNA is going to be translated in the ribosome to give us a target response. That is steroid hormone first messenger system. And that is facilitated because our steroid hormone structure is lipophilic and nonpolar. Just like this yellow part of our cell membrane, it is lipophilic and nonpolar. So likes dissolve, likes steroid hormones will dissolve so easily in the nonpolar segment and cross both membranes, the cellular membrane and the nuclear membrane. Now, the second messenger system is two parts. There is the calcium calmodulin system that is very simple, where the binding of a signal to an extracellular receptor will lead the calcium to influx to the cytoplasm and then bind to calcium calmodulin, bind to the calmodulin receptor. Then the calcium and the calmodulin will mobilize to the inside of the cell, to the inside of the nucleus to get for us a target a target response by making a transcription of the DNA, or it will be able to exert uh, cytoplasmic effects and converting enzymes from an inactive form into an active form. Now, the G protein part is a little bit more complicated, and we're going to spend more time on it. So, the G proteins, there are two effects. The G proteins can either facilitate their target response through phospholipase C or through G protein bound receptor. So, let's begin with the G protein bound receptor. We would have a G protein coupled receptor, GPCR. G for G protein, P is for protein, C is for coupled. That means they're attached together receptor. So the inactive off form, the minus form, will have the G inactive protein receptor attached to a G protein attached to GD as in dog, GDP attached to alpha, beta, and gamma. That is when we are in the off position, we are minus, we are inactive. But once the ligand, the stimulus bind the receptor, there will be a conformational change. And now we're going to be turned from the inactive form to the active form, from the minus form to the plus form, because the receptor is now bound. This was when the receptor was not bound, but now the receptor is bound. So now we're going to move on from the inactive form into the active form, from the minus form into the plus form, from the off position into the on position. So some catalytic changes have to happen. So what happens here, you have how many structures attached? You have the G protein coupled receptor, that's structure 1. You have the G protein itself, that's structure 2. You have GDP, that's structure 3. You have alpha, that's structure 4. You have beta, that's structure 5. And you have gamma, that's structure 6. the binding of the ligand to the receptor will make some conformational changes, and you will never have these arrangements the same way as before. You will never have 1, 2, 3, 4, 5, 6 the same way that it was before in the off position. That is off. So what will change now? What will change is when the stimulus binds the receptor and the conformational change happens, GDP will be converted to a substance known as GTP. So it used to be guanosine diphosphate. That's why they call it GDP. So guanosine with two phosphates. Guanosine phosphate number one, first P, phosphate number two. Guanosine diphosphate, GPP. Now it's going to become guanosine triphosphate, G, P, 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 three P's. So that's an extra structure now that we got. This GTP that we made, GPPP or GTP, this GTP is now going to recruit the alpha, and we will have a complex known as the alpha GTP complex. The alpha GTP complex. And now we will have alpha GTP complex on one side. On the other side, we will have our receptor attached to our G protein, attached to GDP, attached to, so the GDP is no longer there because it's now converted into GTP. So now we will only have the receptor, the G protein, the beta and the gamma, and there will no longer be an alpha position, an alpha subunit. So now we will only have 1, 2, 3, 4. We used to have 6. Now we only have 1, 2, 3, 4. So now what will this alpha GTP complex form? This alpha GTP complex is going to move and activate for us a very important second messenger known as, this is a second messenger. This second messenger is known as adenyl cyclase. Adenyl cyclase was off. It was off. It was not working. It was inactive. Alpha GTP is going to convert it into active form, on. This active on form is going to be made up of alpha GTP and adenyl cyclase. You have GTP, alpha, adenyl cyclase. Then adenyl cyclase is going to perform a very important reaction known as converting adenine triphosphate. So you have adenine with three P's: adenine P1, adenine P2, adenine P3, ATP, adenine triphosphate, adenine PPP, APPP. So adenine triphosphate is going to be converted into adenine monophosphate. So now it's going to look like adenine with one P. After it used to be adenine with three P's. Now it's adenine with one P. This adenine with one P is going to now convert for us protein kinase that was in the off position, an inactive position, a minus position, is going to turn it into a plus, active, and on plus active position, and that is going to convert for us protein kinase that will be activated. And the protein kinase, obviously kinase is phosphorylate. They're going to phosphorylate for us an inactive enzyme and turn it into an on enzyme, and then that will give us the target response that we desire. So you see that there are a lot of cascades, turning an on to an off, turning off to on, turning off to on, turning off to on. But this happened after the configuration has changed with the G-coupled protein subunit. So now when we're going to turn this reaction is not going to go on forever. It has to stop. So there has to be a negative feedback. In order for it to stop, GTP will be hydrolyzed back to GDP. When GDP is hydrolyzed back to when GTP is hydrolyzed back to GDP, now it's going to go back and bind the G protein, and alpha is going to go back to the alpha is going to go back to the initial position. So now we will have alpha, beta, gamma, G protein, GDP, and now we are inactive. There is another form of G protein. That is when the ligand binds the receptor. And when the ligand binds the receptor, now we will not have GTP being GDP converted to GTP and alpha subunit, beta subunit. No, that is one form of G coupled protein association. The second form is when the stimulus binds the G protein, it will activate phospholipase C. And once phospholipase C is activated, what it will do is it will take a fragment from the cell membrane known as PIP2. PIP2 is also known as phosphatidylinositol. That's why there's two of them, because at the 4 position and the 5 position, known as the phosphatidylinositol 4,5 biphosphate. That will be fragmented into 2 second messenger systems. The first one is called IP3, which is inositol triphosphate, and DAG, diacylglycerol. Inositol triphosphate will cause an increase in calcium concentration intracellularly because it will deplete calcium stores from the mitochondria and from the endoplasmic reticulum, and it will cause diacylglycerol to turn protein kinases from the off position to an on position, and that will lead to cellular effects. So these are the 2 G coupled protein receptors that we need to be aware of. How does the activation of a G protein lead to the hydrolysis of GDP to GTP, and how does the binding? So now GTP is going to hydrolyze to GDP is going to hydrolyze to GTP, then GTP is going to bind the alpha subunit, and the alpha subunit is going to go and activate for us adenyl cyclase. Then adenyl cyclase is going to turn for us ATP into AMP. Then AMP is going to turn for us protein kinases, and then these protein kinases are going to phosphorylate for us some inactive enzymes, turning them to active enzymes, and then the active enzymes are going to give us our target proteins. And then we have the other system known as phospholipase C, where the binding of a ligand, the non-steroid hormone, to G protein that is not coupled to an enzyme that is not coupled to alpha, beta, and gamma subunit. It's just bound to G protein. Then that G protein now is not going to hydrolyze GDP into GTP, but rather it will activate phospholipase C, activating phospholipase C from off into on. And then when phospholipase C is turned on, it will convert for us phosphatidylinositol diphosphate, phosphatidylinositol 4,5 biphosphate will be converted into 2 messenger systems, inositol triphosphate and diacylglycerol DAG. Inositol triphosphate is going to increase intercellular calcium concentration by depleting calcium stores within the mitochondria and depleting calcium stores within the endoplasmic reticulum. And increased intercellular calcium concentration is going to exert for us the desired target effect, such as the smooth muscle contraction and the changes in cell secretion. But the diacylglycerol, once activated, once synthesized, is going to convert for us from an off position into an on position. Very important regulatory protein kinases, and these protein kinases, known as protein kinase C, and protein kinase C is going to exert for us important cellular effects. Thank you.